right, let's talk molecular fossils, shall we? And let's bump this up one size, class. Oh, yeah, looking much better. Okay, and we've got a little hemoglobin up here, and we're showing the oxygenation and deoxygenation of the heme group in the center of that hemoglobin. So there you go. And you get the conformational change around it as it is oxygenated. So a heme group actually looks like this. It's a plate-like molecule. It's called a porphyrin ring. Porphyrin. And that's actually true of all of these things. These are all porphyrin rings, and they are commonly used for energy transfer. For example, in animals and plants. It's something we have in common. So yeah, you're closer to a plant than you might think, than you might like to believe. The big difference here is that in animals, the heme group is going to have some nitrogens holding on to an iron atom, and the iron will bond to things or change redox states basically as needed. And in plants, chlorophyll, for example, all the chlorophylls have this sort of porphyrin ring, and then they have a little chain that comes down here. And it's going to have a magnesium in the center, but also coordinated by the nitrogens. In this case, two of the nitrogens are directly bonding, and two of them are hydrogen bonding with their little hydrogens. In this case, all four are needed to bond the magnesium. And this is the thing where light energy is going to be conducted in. It's going to be transferred into the center via excitons and plasmons and that sort of thing. And then it is going to end up exciting an electron in the outer shell, and we're going to start an electron transfer chain. Now, if you were to look in crude oil, right as it comes out of the ground, you would find molecules like this in there. And this is a common one that has a vanadium with a double bonded oxygen in the center. That's weird. But look at the ring, and that is a very familiar looking ring. Now, you can compare this to what we get in both animals and plants. And you can say, oh, it's got this little two carbon chain right there, and the one carbon chain as opposed to the two carbon chain with a double bond and one being opposite. And same thing down here, two carbon chain as opposed to a one carbon chain. The lack of a double bond here may be something that happens during molecular fossilization, because you get alteration of the molecules. And part of that alteration is that some atoms are going to be replaced by other more stable atoms that may not be useful for life. Like life really definitely needs a magnesium here because it's easier to excite those electrons from magnesium and easier to find magnesium on Earth, maybe. And there's a bunch of other uses for it. There's a bunch of enzymes that require magnesium ions. Whereas over here, vanadium, there's some vanadium just lying around in rock and essentially doesn't get used by life in any particular amount, no more than traces, but it can replace magnesium in these things. So the molecular fossil is missing some parts. So this chain down here that went to a long sort of snaky chain for chlorophyll is broken off. We're missing a methyl group and a double bonded oxygen, so we see extraction of those, and we see the nitrogens reacting. They are, it's probably the nitrogens that are actually swapping out the magnesium for, in this case, a vanadium. And if that had been present in the bloodstream of the organism or in the phloem of the plant, I guess inside the cell of the plant, then that change may have happened in life. And if that did happen, you might make a non-functional or less functional version of chlorophyll. So yeah, it's in living things' best interest to keep the machine working, but you get molecular fossilization, you get kind of a lowest common denominator reaction with whatever happens to be around. So yeah, but that's what shows that petrochemicals generally are, for the most part, coming from algae, things that had chlorophyll in them. And they're a mixture of all the chemicals that made up those early plants. So there's a lot of porphyrins in the world. you get porphyrins, for example, in all of these cytochromes. So cytochromes are molecules that are often going to be used for either electron transfer, like energy steps, or also a lot of different enzymes, the ones that detoxify things in your liver. There's a lot of cytochromes there. And those are going to use heme groups, HEME. And yeah, so they are a part of normal electron transfer, but there's a secondary use for those in carrying oxygen, and there's another secondary use as enzymes, as things that have a basically a metal ion at the center. Metal ions can have charges that amino acids just usually don't have unless everything, unless the environment has become charged. And so, yeah, they can do more ambitious chemistry, things with bigger delta G's, basically. All right, here is a little tree of life showing some of the molecules that are specific to each group. And there would also be molecules that are basal, that are shared by some of these groups or multiple groups, things like that. But yeah, there are molecules that are specific. Crenarchial, for example, I think is one of these circular membrane lipids. You'd have to look that up to be sure. A little different than what we have in our membranes. But you can see here up near the end, so a lot of these organisms will have a molecule called squalene, and they will arrange it into different shapes, including sterols and bacteriopanoids, it says. And there are different sterols in the world, and it's usually just the last step that differs. Like all the steps leading up to the last step are conserved and would be right down here at the base. But yeah, you'll get different finishing on the sterols of, for example, algae, a little difference on sponges, and differences on like metazoan animals and differences on plants. Yeah. But you can see fungi and plants. Oh yeah, fungi have their own version, ergosterol. That's just how they all finish their sterols. And these are all going to be components of the membrane that are going to be mostly synthesized by the organisms. But there may be some dietary acquisition as well, like, yeah, when we eat too much foods that contain too much cholesterol. But we make most of the cholesterol inside of our bodies, yeah. All right, molecular fossils. What do we know? Well, these are a lot of types of molecules, and this is like a little overview of the fossil record of each one of these things. So yeah, there are nucleic acids, protein fossils, cellulose fossils, chitin fossils. And it's going down here and showing what's the oldest one where we can definitely show that it exists. In some cases, they're going to be diagenetically modified, but they would appear to be the reaction products that were made from these original molecules. Here's an example. This is it's a Paleozoic, I think, scorpion. And this one's a carboniferous scorpion. And so they're showing here the signal, the X-ray absorption signal for fairly low energy X-rays of chitin, pure chitin, like from a bottle. And this is a scorpion cuticle, which contains a lot of chitin, but not specifically only chitin. And you can see, there we go, that sort of picture. And here is the carboniferous scorpion, which is pretty darn close in terms of X-ray absorption. Now, there are other tests that you can do. And yeah, was it St. Thomas Aquinas that said, beware of the man of one book, beware of the scientist of one technique, I would say. A lot of times it's a money limitation, or people are frightened to do a thing that's too new and outside their comfort area. But at the same time, more information is better than less information. Yeah, for sure. But yeah, this is reasonable evidence that we've got peaks that are right about where we would expect for preservation, probably with a little bit of alteration, of chitin in these ancient scorpion cuticles. Here's another example. Ink from squids is going to be mostly made of melanin. Yeah, there we go. And but you can compare old squid ink to modern squid ink. In modern organisms, the ink is going to be contained in these little, I think they're melanosomes, just little spherical containers of it. Here's a modern little squid, just so you can see it. I think that's a sepia squid. And so here is a analysis of the ink. So this is the ink from modern sepia, and these are the major peaks that you get. You actually do get the same peaks, although the relative abundance of the peaks is a little bit different in the fossil. And this is a fossil that has not undergone much thermal cooking, basically, during fossilization, during all that time, which is pretty awesome when that occurs. So you get a cluster of peaks around 14. This is like peak 14 molecule with an extra oxygen, removed, or with a whatever, it's like a little adduct. Something small has happened to it, but the mass is about the same. And here we go. This is when you heat these things during fossilization or potentially after fossilization if you want to do that. You'll see a shift here. You tend to go away from these sort of small molecular weight products. They tend to link up into larger chains just because there are reactive atoms in there. The nitrogens in these are particularly reactive. The oxygens are fairly reactive as well. And they will reach out and they will bond to other molecules, other melanin molecules, but also anything else in the cell. All the carbohydrates, all the lipids, all the proteins end up linking to each other once they're in the rock because there's no more consequence in terms of life, and because these are little reactive molecules that are all clustered together. And so, you get those things forming. And then shown here at the end, this is the very small but detectable signal from steranes, things like cholesterol, that are there. So here are some molecules. This is showing what happens to cholesterol over time as it breaks down. You'll get maybe the sponge version of cholesterol, which is a 24 isopropyl cholesterol. And that's going to get modified. We're going to remove this hydroxyl that's over here because remember, hydroxyls are very reactive. Those will get pulled off. And we will rearrange that electron that was there. It's going to go down and become part of a double bond here. And then from there, if it is modified further, usually the next thing that happens is that these carbons end up cross-linking with something else, like with the rock around them, maybe with some of those aluminosilicates. So silicates are silicon dioxide, which has oxygens, and those oxygens want to react too. So the rock is reacting with the biology is reacting with other biology is reacting with the rock, and it's forming cross-links and bonds that you wouldn't get in a living organism, but you totally get during diagenesis. So yeah, diagenesis is essentially kind of a lowest common denominator process. Whatever the environment is, if there's too much of something, it will affect all the molecules that are there, and they will react in what is probably the lowest energy, easiest possible way, given the current density, concentration, amount of heat in the environment. And so it's a little hotbed for chemical reactions. Yeah, fossils are chemistry. Chemistry is underlying fossilization. That's a weird way to think about it, but why not? Anyway, what they can do with this is they can start to look back and see, all right, can we detect the original molecules? Because some of them don't get destroyed. Can we detect versions of those, like steradienes, which, based on this branchy structure up here, can only have come really from something like this? And can we detect some of the breakdown products of this? And you actually find those all across this area. So here's the early Cambrian. Here is the Ediacaran. And those periods do appear to show sponge like biochemistry was present because you do get these little steridines and styrene isomers in sediments. And so that indicates that something biological was probably making them. Because if you look earlier, like super early, you do not find these things. They do not occur, as far as we know, in any abundance naturally. These are particular molecules made by life as we know it. And if you think about it, if we're searching for life as we know it on other planets or something, there's potentially a little spectral signal from some of these molecules that would really stand out and would be some of the things that they have those Mars rovers and Mercury probes go look for. So, yeah, interesting. Sometimes it's hard to get a good picture. Sometimes you have to get really close. Sometimes you have to concentrate that stuff. So we have rovers drilling into rocks and examining everything that's coming out of there in the hope that we will actually find something unexpected. Yeah. What happens to DNA? Because everything that was alive probably had DNA in it, because everything that's alive now has DNA in it, yeah, and RNA, yeah, for that matter. Yeah, so where does that go? Well, here's the process. So the first thing that tends to happen is that you're going to have all the nitrogens reacting. And there's a nitrogen right here that's trying to do too much. It's holding on to 1, 2, 3 different carbons at the same time. That's a lot, wrangling 3 carbons. So you'll get this nitrogen reacting with something else, and that's going to break this bond, and you'll have something like an oxygen, for example, coming in here. So the first reaction you get is depurination. All the purines, like guanine and adenosine, are going to react and be gone. And you'll end up with these A basic sites. And you can say, well, that was probably a G or an A, but the chain is still intact here. And the C's and T's last a little longer. Or rather after that, what you'll get is you're going to get, and here we've actually broken the deoxyribose ring. You'll get an attack here. This oxygen is going to give back its electron, and it's going to go with phosphorus, and it's going to be an oxyanion down there, and it'll probably end up reacting with something in the rock. And so you're going to get a phosphate that's going to go on to form some other mineral, but you're going to have a bunch of phosphate in that mineral because it came from DNA, and DNA's got one phosphate for every base. So yeah, you'll lose the side chain, and then you'll fracture the actual chain. And it's thought that there should be no sequenceable DNA after, in the best possible case, about 6 million years. That's probably the outermost limit. There are studies that go back at least 1 million years that have sequenceable, really small chunks of DNA, because you get some of this depurination and some of this fragmentation. But yeah, still readable and understandable to some extent. And we'll cover that toward the end of class. Another thing that happens actually very fast, even in like archaeological samples from 500 years ago or something, is you'll get deamination. So there's an amino group right there, a little amine group, and you'll get hydrolysis at this bond. You'll get basically an oxygen attacking and taking this over, and you'll get the NH2 taking back its electron, or no, maybe just getting kicked off, yeah, because nitrogen doesn't care. It'll take on a positive charge. And so you'll get replacement here, and which basically turns a cytosine, C nucleotide, into a uracil, which is like the U or equivalent to the T nucleotide. And so that's a change you see in ancient DNA, is that a lot of things that probably should be C are turned into U instead of T. And if you have a sophisticated enough technique, you can either use an enzyme to convert all the U's back into C's, like reaminate them, putting the amine group on, and then you can sequence it, or you can just read it out, compare it to living things, and say, oh, what do you know? We got a whole bunch of extra T's or U's here, and it looks like in everything else that's alive, that should be a C, and so you can say, yeah, all right, it was probably a C, so we'll either ignore that or provisionally change it, something like that. Okay, hemoglobin. porphyrin rings carry electrons. Nice. You find this in vertebrate animals for sure. I think some chordates. But in a lot of invertebrates, arthropods in particular, I believe some mollusks, annelids, things like that, you will instead get hemocyanins. They tend to produce greenish or bluish blood. And they're going to have copper in there, and copper is going to carry an oxygen molecule instead of the iron carrying an oxygen molecule. But you know what? It works the same way. They do the same job. And so around here you can see the protein that is helping to hold on to the copper and protect it on its journey. Yeah. Hemoglobin just has 4 of these globin proteins, each of which has a little heme group. And each of those heme groups can potentially get an oxygen added to it. Although I don't remember if you have more than one oxygenated at a time. Yeah. No, that's a physiology question. This is not a physiology class. Go ask your A&P prof. Here are heme molecules, just like in the documentary Jurassic Park. Yeah, so here's a little mosquito fossil, and this mosquito appears to be female based on the appearance of it and relation to modern mosquitoes. You can tell the difference. And here is the heme signal you would get from a pig. And this is, so it's a mix of mass spectrometry, that's the time of flight, and SIMS, which is I believe it's an X-ray photoelectron sort of spectroscopy. Think of it as a way to shoot energy at a substance that will read out the atomic content or molecular content. And so here we're comparing the heme signal from heme from a pig to the signal for heme from a fossil mosquito. And darned if those two don't look pretty similar. Here it's zooming in on some of these little peaks. So this is this one little peak. And you see they even have the same little double peak right there. This mosquito, this super ancient mosquito, which I don't remember the age on this. No, they say it in the paper. But I would imagine that is Eocene, just because you get a lot of good mosquitoes that look like that from the Eocene. Yeah, but whenever it's from, that appears to be heme in there. Now, there's probably no DNA in there, so we can't just splice in a little frog DNA and, bring whatever it was that she was drinking back to life. But, yeah, there are molecular traces of whatever it is that she was eating there. And maybe someday we can find all the parts of these molecules that are still present and figure out how to read them out and actually get sequences from some of these older organisms, because the atoms don't go anywhere. They just break certain bonds, react with other things, and it's more a product of the way we do sequencing that makes these unsequenceable. It's not that the atoms wouldn't be there and you couldn't figure it out if you had a complete 3-dimensional atomic map of everything in the fossil. So that costs a lot of money, but that's where the science is headed, I hope. Yeah. Shown and see, this is something else. Let's see. Spectrum from the matrix. Oh, this is what the rock looks like next to the mosquito. So yeah, in the abdomen of the mosquito, there's heme from blood because she was drinking blood. Around the mosquito, there is not, because, yeah, of course. Here is showing it's an elemental scanning map, scanning electron micrograph in backscattered electron modes. We're looking at electrons that are being backscattered, like coming back to the detector because they're running into big, heavy atoms like iron, for example. And so if you tune the electron beam, or if you know the amount of energy you're expecting them to bring back, you can say, all right, where's the iron in this sample? And bam, there it is. This little blob right there is a little tiny blood stain, probably. Isn't that cool? Yeah. And they're showing its elemental composition. So molecular fossils, altered molecules. And this can be any organic molecule. The ones that have been used most in these studies are things like cholesterols, all these sterols, and some other molecules, carbohydrates, things like that. Occasionally DNA, although the reactive nitrogens and phosphates tend to make that a pretty dicey proposition as you get back to older fossils that are more interesting, frankly. How do they differ from biomolecules? Well, the nitrogens are all going to go as free agents and react with something else, whatever they want to react with. They're tired of bonding to carbon to keep you happy. They're going to do their own thing. So good for them, right? Yeah. What are some ways that porphyrin compounds are used in plants? For example, chlorophylls. Yeah, a lot of those energy transferring compounds. In animals, yeah, you get heme, you get cytochromes, stuff like that. What changes would you expect to find in DNA? Look back at that slide because there's a couple of changes. A basic deamination and then just fragmentation between each base at the phosphate bond. What are some functions of melanin in extant organisms? We get that in squid ink, we get that in skin, we get that at the back of our eyes because we're vertebrates, and that's what vertebrates do. Also, some invertebrates. What's the origin of petrochemicals with vanadium porphyrin rings? Probably algal, some sort of plant. It looks like a modified version of a chlorophyll, basically, as shown in slide one. Like, ooh, yeah, let's all look back for slide one. Okay, I think that's it. That's good. Thanks a lot.